As we continue on our study of the cell and its internal workings, we have now come to the all-important endomembrane system. So this is the majority of the cell is composed of the endomembrane system. And it's the traffic cop. So it regulates protein traffic and performs metabolic functions in the cell. So the components of the endomembrane system, there are many. 1, the nuclear envelope. 2, the endoplasmic reticulum. 3, the Golgi apparatus. 4, lysosomes. 5, vacuoles. And 6, the plasma membrane. And these components are either continuous or connected by transfer vesicles. Okay, and the nuclear envelope and endoplasmic reticulum are continuous. So the outer membrane of the nuclear envelope becomes the endoplasmic reticulum, and you'll see a picture in a minute. And then vesicles, which are little sacs, link up the rest of the components. So vesicles linked. Okay. It'll make sense in a minute. So we'll start off with the endoplasmic reticulum, which is a biosynthetic factory, apparently. So the endoplasmic reticulum accounts for more than half of the total membrane in most eukaryotic cells. As I just said, the endoplasmic reticulum membrane is continuous with the nuclear envelope. And there are 2 types of ER. One is the smooth ER, which is smooth. It does not have ribosomes. And the other one is the rough ER. And so the surface is rough because it's studded with a whole bunch of ribosomes. And ribosomes, if you recall from the previous screencast, are the site of protein synthesis. So they make proteins. All right, so here's a picture of the nuclear envelope here, the dark purple, and you can see how the outer membrane of the nuclear envelope is continuous with the endoplasmic reticulum here. It's continuous with the endoplasmic reticulum. Okay, and then you see this ER here that's studded with ribosomes, all these little bumps. That's my rough ER. Okay, and then out here you have endoplasmic reticulum with no ribosomes. That's a smooth ER, and you see the rough ER is more kind of like flattened sacs, like pancakes, and the smooth ER is more like tubes. Okay, so tubes there, and then flattened sacs. Okay, so what does the ER do? Oh, there's a live picture. This is actual ER, and you can see here rough ER studded with all these ribosomes, and here is the smooth ER. All right, so let's look at the smooth ER first. Smooth ER synthesizes lipids. It helps to make more membrane. It metabolizes carbohydrates, so it breaks down polymers of glucose. This is handy. It detoxifies drugs and poisons. So if you take an aspirin, it gets rid of it. It gets rid of the toxic substance out of your body. It metabolizes it out. If you, and again, that's any drug, alcohol, tobacco smoke, like any of that stuff, all gets the smooth ER gets rid of all of that. And it also stores calcium. And calcium, you guys probably associate with having strong bones. But calcium is also a very important coenzyme for a lot of metabolic reactions. And it's very important for muscle contraction. And it needs to be around for lots of cellular functions, not just for strong bones. So go drink a glass of milk. All right, so that was the smooth ER. Now we've got the rough ER. Because the rough ER has ribosomes, it, of course, plays some kind of role in making proteins. Okay, so the rough ER has bound ribosomes which secrete glycoproteins. So that's a protein that has a carbohydrate attached to it. Proteins covalently bonded to carbohydrates. Okay, then it takes those glycoproteins and distributes them in transport vesicles. So the protein is inside and it's surrounded by this little membrane, and then that kind of also gets moved around. And because of moving membranes around, it's a membrane factory for the cell. It can also make membrane and distribute it throughout the cell. We're going to put all this together at the end. So a separate part of the endomembrane system is the Golgi apparatus. Personally, my favorite organelle. It is a shipping and receiving center. So it's basically the post office. Post office. Okay. So it receives vesicles with carrying proteins from the rough ER and then packages them up, sorts them out, and then ships them off to where they need to go. Okay. The Golgi apparatus consists of flattened membranous sacs called cisternae, and it basically looks like a stack of pancakes. Okay, you'll see in a minute. Functions of the Golgi apparatus: it receives products from the ER and modifies them. It can manufacture some macromolecules, but mostly it lets other people do that. It sorts out the materials it receives and packages them into transport vesicles and then ships them out to where they need to go. So it's a post office. Here's a picture of the Golgi apparatus. You see, it's like this flat stack of pancakes. Okay, here's a vesicle coming from the rough ER, and you see there's little proteins inside. That side fuses with the cis face, or the receiving side, and then those proteins, as you see, actually I'm going to erase this mess here, move along and get sorted out and processed, and then get packaged into other transport vesicles, which get shipped. So here's another transport vesicle, and that's going to get shipped to wherever its next destination is. And the trans face is the shipping side of the Golgi. Okay. Moving on, lysosomes are another organelle. They're basically little digestive compartments. So a lysosome is a vesicle that has enzymes in it that can digest macromolecules. So enzymes that help catalyze the hydrolysis, they're hydrolytic enzymes, help catalyze the hydrolysis of macromolecules. So it breaks down our proteins, it breaks down our carbohydrates, it breaks down our nucleic acids. Okay. And there we go. Hydrolyzed proteins, fats, polysaccharides, nucleic acids, breaks them down into parts. Okay. The lysosomal enzymes work best if there's an acidic environment inside the lysosome. It creates this little environment so that the proteins inside have the correct structure so they can have their correct function. Some types of cells can engulf another cell or bits of food by a process called phagocytosis. We're going to talk about that in the next chapter, chapter 7. That will give you a food vacuole, and then the food vacuole will fuse with the lysosome, and then the digestive enzymes will break down whatever was in the food vacuole. Okay. Lysosomes are also used to recycle the cell's organelles and macromolecules. Okay. So if you have damaged macromolecules or damaged organelles, they can be broken down into parts by the lysosome and then recycled back into new organelles. So let's see how this looks. So here we have food. So here we have food, yummy, being ingested into a little food vacuole right here. Okay. The food, here's our lysosome. It fuses with the food vacuole, and the digestive enzymes, the little blue blobs, break down the food and digest it. Okay, so that's one function of the lysosome. We have another function of a lysosome. Let's say we have some damaged organelles. We have a damaged peroxisome, a damaged mitochondria, and they'll be packaged up into a vesicle. That vesicle will fuse with a lysosome. Okay, and then the digestive enzymes will break down the damaged organelles into parts, and then they can be used to rebuild new organelles. And look here, and here is what an actual mitochondria looks like. There you go. And the last thing we're going to talk about are vacuoles. And vacuoles perform a wide variety of roles. Okay, plant cells or fungal cells can have several vacuoles, and they come from endoplasmic reticulum and Golgi apparatus. So there can be food vacuoles, which are formed by phagocytosis. Contractile vacuoles, these are kind of cool. Freshwater protists used to pump excess water out of cells. And then central vacuoles, which are mostly for storage. And central vacuoles are kind of the most popular of the vacuoles. So if you have a look here, here's a plant cell, and you'll see it has a very large storage component, the central vacuole there. It's used to store water and nutrients and things like that. Water here, this right here, this is a chloroplast. Here's another chloroplast. Here's another chloroplast. So chloroplast is where photosynthesis occurs. And photosynthesis requires lots and lots of water. So the plant needs to have water stored up in its central vacuole. Okay, so that's the endomembrane system. Quick review. So it's a complex and dynamic player in the cell's compartmental organization. And let's go to the next slide, which kind of brings it all together. So you see you have the nucleus, and the nuclear envelope is continuous with the rough ER. The smooth ER is over here, dedoxifying things and making lipids. The rough ER then connects to the Golgi apparatus by, here we have some rough ER that's made some proteins. They get packaged up into a vacuole, or sorry, a vesicle, not a vac. A vesicle, transport vesicle, and it brings the contents to the cis Golgi. They fuse, and then the contents get kind of sorted and packaged out through the Golgi. And then a couple things can happen. They can get packaged up into another vesicle that takes them to the plasma membrane. It can fuse with the plasma membrane and dump its contents out of the cell. Or they can get packaged up into a vesicle, and that vesicle could, say, become a lysosome and then do its little digestive thing throughout the cell. So you see it's all kind of connected. So if it's not physically connected, like the rough ER and the nucleus, they're connected by these vesicles. Okay? And that is the end of the endomembrane system.